Okay. All right. Welcome to your new recording of Lab 1. We have covered basically all the different cell types of the white blood cell system, and we've given some examples. Now, moving forward, let's go down to this table, right? The erythrocyte. And the erythrocyte basically is a fancy word for red blood cell, right? Red blood cells. And what is their function of red blood cells? Does anybody know? Chat me something.
To transport.
Transport oxygen. I just heard transport. Yeah, transport oxygen. Now, another interesting thing about red blood cells is that they do not have a nucleus. That'll be a test question eventually somewhere down your academic careers. All right, I've had it. What is an anucleated cell, or what cell type in the body does not have a nucleus? All right. And chances are one choice is going to say a red blood cell. Red blood cells, as they mature, the bone marrow will pretty much suck out their nucleus. So they are released either with a nucleus that's getting disintegrated or getting pulled out, or pretty much all anucleated. Now, if you were to have some kind of hemorrhagic loss, a hemorrhagic loss of blood, and you were low on blood, and the bone marrow senses that you were low on blood, like it said with the white blood cells, it will release a bunch of red blood cells, immature red cells. And you can always tell what the immature red cells look like because they have nucleus. They have a nucleus in them. So that's babies. Babies are like this when they're born. Sometimes if they're struggling to breathe, or they're premature, they'll have a lot of nucleated red cells in them. And I can always tell that because you can see the little nucleus right in the middle of the red cell. And, eventually what happens when it gets released out into the peripheral circulation, the spleen will help pull the nucleus out of the red blood cells. So once the nucleus is removed from the red blood cell, it's able to carry oxygen and do its job. Red cells live anywhere from 90 to 120 days. All right, somebody emailed me. I want to make sure. Okay, I'll have to get back to her. All right, so neutrophils we've kind of already talked about. Those are in response to bacteria. Basophils do all kinds of different things. Basophils basically are involved in more virus, sometimes virus responses. Kind of like well, that's all we're going to say. And then EOs are allergy or parasite infection response. And then lymphocyte is viral response. And then monocytes, also want to tell you that monocytes are, because I told you that they consume infections, they are phagocytes, okay? Neutrophils and monocytes are phagocytes. And what I mean by that is that they phagocytize their target, okay? Whether it be like a gram-positive cocci, staph infection or staph bacteria, staphylococcus bacteria. They will consume that, and then they will destroy it within the cell. If that makes sense, as I'm using my hand here to gesture what a phagocyte is. Does that make sense to everybody? I wanted to add that in. It's very important that I try to add in the vocabulary so you can connect the dots. I can't just tell you that they eat something. No, you're not going to learn it like they eat something. You're going to learn it that they're a phagocytic cell, a phagocyte. That's the keywords I need to make sure that we connect the dots on. Okay, I'm going to pull down this chat again. Okay, everybody's good, it looks like. All right, blood typing. All right, this is getting into this is some elevated material, and we're going to go back on the immune system because this is transfusion medicine. All right, this is what happens when this is what happens when baby and mommy and the baby or fetus and mother have different, different blood types can cause problems. It's called HFDN, hemolytic fetal disease of the newborn. We will touch on that. If you have lost blood for whatever reason and you need blood, this is what we draw upon to make sure we give you the right blood. If this is a traumatic event, kind of like a triage situation, and we can only give you O negative. We're going to talk about why we give people O negative and so on. So before we do that, be able to determine the blood type from the sample and be familiar with the term. So I'm not sure if we have these kits at the house. I'm not sure how Dr. Davis did those. I likely think that none of you all have these kits. So we are going to probably talk about this in a conceptual manner. If we were in laboratory, we would be doing this and you could kind of see what the reaction looks like. All right, so what are antigens and what are antibodies? Those other two words, I'm not sure if we're going to touch on those much. I really want to focus on antigens and antibodies, okay? Antigens are in reference to red blood cells and what pretty much is us. And what I mean is that, like, what is self? And antigens in terms of, well, I probably should introduce this as just what it is in immunology, then we'll go to what it is for self. All right, so one way that the immune system responds to certain foreign bodies is through their antigens, okay? And what I mean by that is that antigens are like molecules, glycoproteins, proteins that have sugars on them. You don't have to know that. But antigens are found on everything, okay? It's kind of like an identifier. Think of it as like a person's face. You identify somebody through their face. A good example would be like, this is my antigen. This is my face. This is how you identify me as Drew Mattmiller, okay? So now, antigens on bacteria, this is how white blood cells can identify these bacteria through their antigens, okay? All right, now we've already kind of talked about antibodies. So what antibodies are is also another molecule that either will be released into the circulation or will be found on the surfaces of white blood cells to attach to the antigen, all right? Antibodies' focus is to find the antigen and attach to it, all right? And then how they attach to it will signal other molecules to come in and degrade it, all right? It's kind of like a tagging mechanism, if that makes sense. This is when I wish I could see you all in person and I either like see confused looks or I see like people shaking their heads. So try to stick with me because you want to get these words right and you want to spend time with this because blood bank can be, even for me, it can be challenging material. All right, so that is what happens when you have, kind of like cops and robbers, right? You have the robbers as the bacteria and you have the cops that are trying to catch the robbers, if that makes sense. All right. The antigens normally in the body are going to be found on bacteria. They're found everywhere, but we won't recognize our own self antigens. Okay. If we do, that's called an autoimmune disease. All right. We've probably heard of that. Lupus is an autoimmune disease. Multiple sclerosis, they think, is an autoimmune disease. Myasthenia gravis, perhaps, is a autoimmune disease. And the point that I'm trying to make is that we don't want to recognize ourselves, right? We recognize antigens from foreign bodies, from foreign things that are in our body that, like people have grafts put in, tissue grafts. Sometimes when we have transplants, this is a big deal. When we transplant an organ, we have to lower the immune system because in all likelihood, the transplant is from somebody else's body and therefore they have different antigens and then we have to pretty much like put the immune system on ice so they do not respond to it and essentially kill it. That's what the immune system does. It kills things, and we don't want it to kill our new liver. So there's things that we can take to, to lower the immune system. All right. So that is pretty much a quick introduction of antibodies and antigens and the cat and mouse game that it is. Antigens try to elude the antibodies, and then antibodies try to tag the antigens so the immune system can come and kill it. Okay, so we're good there, right? All right, good. All right, let's talk about blood bank. Let's talk about blood types. On our red cells, we either have A, B, AB, or O antigens on our red blood cells. All right. They are also called codominant. You don't have to know that. What I mean by that is that you get one from your father and you get one from your mother. All right. These are genetics. And depending on what genetics you have, they come together and you will have a blood type of either A, B, AB, or O. Likely, the interesting thing is it's all based on like population. Chances are in the state of Kentucky, you are going to have 45% A positive. All right. You're going to have 15% B positive, another 15% B negative. And then O, I think, is also another like 40% or 45%. And then AB is like maybe 5%. So it's kind of funny because when sometimes we have to get blood from like Pennsylvania because Pennsylvania has a large population of O positive, right? If we don't have O positive. And what they do is they put these blood units on helicopters and they move them everywhere. It's kind of interesting, especially if they are short dated. The blood can only last X amount of days. I think it's maybe 3 weeks, maybe. I can't remember. So now let's go back to the table with the blood types. A antigen or A blood type has A antigens. Okay, that's going to go for all of them. Let's just do those first four. We'll talk about RH next. So B is B antigens on red blood cells, and then AB is going to have both of them, both antigens on the red blood cell, and then O. When I say O, what it really is that it doesn't have an antigen at all. So think of O as like a zero, like they're missing that glycoprotein. It's just they have antigens, but they're missing the key thing of that antigen that makes it that antigen. So just think of it as like no antigen at all. If that makes sense. Okay. Now, antibodies. I don't want to go too fast because I think this is some elevated. This is, we're starting off pretty heavy here with blood types. Now, A blood type. If you have A blood type, you are not going to make A antibody, because if you think about it, A antibody, if this is my antigen on my red blood cell, you're not going to make A antibody, because what's going to happen is that A antibody is going to attach to A antigen, and we don't want that. We don't want a bunch of stuff clotting off in our vessels, right? So we're not going to make A antibody. What we will make is B antibody, all right? And that's just natural occurring, B antibody. All right. Now, for B antigen, we have, or B blood type, we have B antigen. And then for B antigen, we're going to make A antibody that's naturally occurring. All right. So for blood type AB, we have both the A and the B antigen on our red blood cells. And then for that antibody, we do not make any antibody. All right, so no antibody for AB. Now O, maybe you're picking up on this, is the opposite. So we do not make an antigen for O. However, we make both AB antibody, A and B antibody for O. All right. This can be confusing, y'all. It's probably a good thing we did record this so you can kind of go back and revisit some of this material. Okay. So I'm just going to check this chat. Okay. So we're good. All right.
I have a question.
Okay. Lay it on me.
So basically it's opposites. If you have A blood type, you have an A antigen and a B antibody all the way
For,
until you get.
all right. Hold on, Ryan. You said for A blood type, you said?
Yeah, it's basically just opposites, correct? If
Correct.
you have an A antigen, it's a B antibody.
Yes. Yes.
Okay.
Because you don't want these antibodies and antigens coming together. Now that would be like a pathology, like something's going on, like some kind of pathology is going on.
Okay.
I.e., what I mean is like autoimmune disease. That's what happens when the immune system starts to recognize its own self, its own antigens. Then we have a problem. And there's diseases out there. Lupus is the big one. Okay. All right. So now, when we talk about positive and negative, you might have heard me say O negative or O positive or A positive. The positive part is the Rh antigen. All right. There's probably, I think my blood bank instructor told me one time, because she's a specialist in this field, she says there's hundreds of antigens on red blood cells, hundreds of them, and they're all different types. And there's only a handful of them that are serious that can elicit an immune response. And what I mean by elicit, I mean by pretty much poke the bear, wake up the immune system and get it attacking something. A, B, and O are the big ones. The second big one is the Rh antigen. Okay. That's why we include it, because the Rh positive means that you have this antigen. You are positive for the Rh antigen, and you have to account for that. All right. So jot that down. Rh positive means you have this Rh antigen. If you're wondering how I got its name, it came when they were studying and working out all this in the 50s. The Rh, they used monkeys to do this, and they used the rhesus monkeys, the Rhesus, rhesus monkey type of species, and that's how they found this antigen, was on the rhesus monkey. So it's Rh. That's how we, that's what we call it now. Okay, so and then you have Rh positive, and then you have Rh negative. Rh negative means you do not have the Rh antigen. All right. Okay. Rh is it's uppercase and then lowercase, Ryan. And then D, I think if we talk about what ROGAM is, we may talk about D, but we probably won't touch on D too much. I think that's probably getting too involved there. Okay, so I think that's all we're going to do with antibodies, blood types, A antigen and antibody. I don't want to confuse you all too much. If we were to get into transfusion, that is something completely different. If let's say if I'm A positive and I'm losing blood, that obviously the first match for me would be to give me A positive. All right. Now the reason why we can give somebody O negative is because essentially you're just getting red blood cells. Because if you remember what I said, O blood types do not have any antigen on them. They just have red blood cells. So we're not going to, because see, if I'm A positive, if you remember, I'm going to have a naturally occurring B antibody. All right. So if I have this B antibody circulating around and you give me O negative, then nothing happens. Okay. However, if let's say you screw up or somebody screws up on my blood type and they transfuse me with B blood type, then that means that new blood coming into my body is going to have the B antigen. Now, if you remember, I told you since I'm blood type A, I have the B antibody. So guess what happens? My B antibody finds its partner in the new blood that has the B antigen. It connects, and then it'll what we call is hemolyze that new blood. All right, pretty much cuts it in half and renders it useless. And not only that, but it'll wake up a bunch of other stuff and can cause problems, and eventually it'll kill you if they don't do some kind of acute response very quickly. It'll kill you. In babies, it's the stakes are higher because it's a much smaller body. If we had to transfuse a baby, and sometimes we do have to do that. Okay, so that's just an example of what happens when you transfuse somebody and something were to go wrong. But you guys have this on recording. You can listen to it again if you prefer. Okay, let's go down to part C. All right, let's get rolling here. White blood cells. So the normal ranges for white blood cells on a complete blood cell count that I work with is anywhere from 5 to 10,000. Okay, millimeters. No, I'm not going to embarrass myself now. I used to know those. It's not millimeters per deciliter. It's some kind of volume. I'm not gonna touch it, but please review those units. You will probably see those units a lot. We usually deal in millimeters per deciliter. Let me check this chat. Okay, sure, Derek, I'll go over it. The RH blood type is what is the positive and the negative part. So when I say I'm A positive, that means I have the A antigen and I also have the RH antigen. Okay. And the RH, if I am A negative, then I have the A antigen, and then I do not have the RH antigen because I'm negative for the RH. The negative and the positive are only talking about the RH. It really doesn't matter. The big deal with RH is when mothers tend to have different blood types in their baby, because what kills the baby is the RH antigen. That is what does the harm on the fetuses. It can be a big deal. It won't necessarily kill the first child, but as the mother becomes sensitized, becomes informed of the baby's blood, it'll build up antibodies against it. So I don't want to go, it's kind of cool. I do want to kind of touch on this. So if mom is A blood type and baby is B, sometimes what happens is that baby's blood will leak into mom's circulation through the umbilical cord, right? Well, mom's immune system will recognize this foreign blood, i.e. baby blood, and it'll build antibodies up, right? It takes a while. It probably doesn't do too much harm to the current child. Let's say mom, after a year and a half, 2 years, becomes pregnant again with her second child, who is also B blood type. All of a sudden, mom's got this loaded gun of B antibodies. And what happens is that there's one type of antibody that can cross the placenta, and that is IgG. That's a subclass. We'll talk about that later. These antibodies can cross the placenta and attack the second baby to the point that it'll succumb in the womb and you'll have a stillborn. Luckily, we have RhoGAM, which is something that we can give mothers to prevent this. RhoGAM is just prophylactic antibody that pretty much renders any antibodies made up against the baby useless. we really don't know to this day how it works. We just know it works. We don't know the true mechanism of how ROGAM works, but what it is that we give these mothers a shot of it, and it'll kind of it'll calm down any antibodies built up against the baby. This will sometimes, when it gets serious, is when people have not had prenatal care. There has not been any prenatal care, and usually when this happens, it has a name. It's called HFDN, hemolytic fetal disease in the newborn, and it'll happen in the 3rd trimester. And usually, you probably have a matter of days until you have to act. And what I mean is, you probably want to induce and take the baby early. But it could be avoidable, but it's also a poor outcome if it does happen. All right, so sorry if I got off on a tangent there. Yeah, Courtney, you got it there. No, it's not per mole, Clarissa. That's more like general chemistry. This is it'll come to me after this. You can probably Google it. It's a volume. It's something that you don't need to focus on too much. It'll come to me. It bothers me that I can't remember it. All right. So if you are A positive, you have the Rh antigen, and vice versa. Yeah. If you are A negative, Rh negative, then you are A negative. Correct. Okay. Yeah. Irina, yes. I think, please go over that. Scott Davis and I think it's Scott Davis and maybe Dr. Sharp are doing the Bio 139 spotlight sessions. I do encourage you all to do that. So if it's unclear here, please sign up for a spotlight session on that. Okay. So red blood cells are anywhere from 4.6 to 6.2 million. So you have to realize the magnitude of difference here. White blood cells are in the thousands. All right, thousands. There's not too much of them. Red blood cells are in the millions. I just want to clarify that. All right, let's talk about leukocytosis. Leukocytosis, when you try to break down these medical terminology words, osis means there's a problem. Likely what it means is that there's some kind of inflammation of something. Something's getting bigger because something bad's going on. It's real plain terminology, so osis means that there's some kind of pathology. Leuko means white, and then cyto means cell. So put it all together, it means you got a very high white blood cell count, or a very high leukocyte count, over 10,000. We had a patient in last night come in who had 434,000 white blood cell count because he had lymphoma, and I think he's gone off of his treatment. He's 79 years of age, and at this point, it may be like he may be a palliative care patient. But I had to work him up, and he was already diagnosed with lymphoma, but we still have to look at the cells and kind of see what they look like and how hairy they look. So in that case, he had leukocytosis. All right, penia. Penia means small, or lack of, or lessened. So leuko means white. So leukopenia means that you have a very low white count. Okay. Polycythemia. Poly means many, and then emia. When you have osis and you have emia basically deals with the blood system. All right. So osis could be anything outside the blood system. When you hear this emia, like leukemia, that is pertaining to the circulation. And so this means that you have many cells in the blood. So what that translates to is that you have a high red blood cell count. All right, a high red blood cell count. Anemia is if somebody's anemic, that means they've lost blood. Okay. All right, we're going to dig into some of these case studies. Okay. Now we're going to do these together because we don't. It doesn't look like we have the kit. All right, so I will scroll up and down. I want to see how many I have here because I need to manage my time. All right, I will not touch too much on the pig. If we're scrolling through on page 6, 7, and 8, these are memorization. This is something that we will pull from that you guys should have the outline of what they look like. There may be pictures. I may be able to take pictures of what the answers look like when I go into the lab. I had to do this last semester was for some folks. And I will send out the pictures of the answers. I think there are answers somewhere, but this is more like memorization and flashcards, and it's just something that we're going to have to touch on now. So I'm not going to spend too much time on that. What I do want to spend time on is the case study at the very end. We'll kind of touch on that. I don't want to go over too much of it because I want you to work through the anatomy of these. But let's scroll back up to the blood bank case study. Okay. All right. So on page 4, we have an unknown simulated samples for Mr. Smith, Mrs. Jones, Mr. Green, and Miss Brown. We have, if we're in the blood bank and we need to type this blood up, we have these, what I'm going to call reagents, which is like these little factory-made serums that help tell us what in the world this blood is. This is kind of how we type people out if they were to need blood. Okay. So let's go down to the procedure, which we're going to skip because we don't have it. Now, how we do this, how myself and my colleagues do this in the lab is that we look for that antigen antibody reaction. Okay. And so when enough of this antibody antigen come together, it's going to what we call agglutinate. All right. And that means clump up, and large enough clump to where we can see it. Right. And it's going to be like a little like dot of like, I don't know, like a little clump. It's like a little clump. And those are like the red blood cells interacting with each other because they have the same antigen and antibody and they recognize each other in clump. In the body, we do not want that. That's bad. We want everything to run together and to not recognize each other and then circulate freely. All right, so we kind of have to understand what in the world's going on in the body, and then what in the world are we doing when we're trying to type this blood out. All right, but what it basically boils down to is we want it to clump in the lab, because that means, okay, if we use this serum, if we have this antibody B serum that we have from the factory, and we mix it in with somebody's blood that happens to be a blood type B, it's going to clump, right? Because if we have this factory made antibody B and we have a B blood type and we mix it, it's going to clump. So we know that person is B, has a blood type of B. So that's what we try to give. If we have to transfuse them with blood, we would give them B blood. Okay. Because if you have B blood, you don't have in your body, you do not have B antibody. You will have A antibody. All right. That's kind of a elevated concept there. All right. All right. So let's go down to the graph. This is when gonna have to change gears, and this is when we are talking about being in the clinical lab, because we're gonna be the blood typers. We're gonna be the transfusionists here. Okay. So we said Mr. Smith let's say Mr. Smith has A blood type. Okay. Let's just write down a big A positive right below Mr. Smith, right? Because in the lab, we wouldn't know. We're going to figure this out if we were in the lab, but we don't have the kit in front of us. So we're going to identify Mr. Smith as A blood type. Okay, so we take Mr. Smith's blood and we mix it with some factory-made antibody A serum. When you see anti-A or anti-B, when you see that prefix is anti, we're talking about the antibody. All right, so we have anti-A serum, right? And we have Mr. Smith, who is an A blood type. So does anybody want to tell me what we're going to get here? Are we going to get clumping or are we not going to get clumping? That's basically what we're looking for. Yeah, good job, Raymond. We're going to get clumping. Does everybody understand that? It's just one response. Okay, yeah, Derek, yeah, clumping, okay. All right, so if we take the factory-made antibody B serum, who has the B antibody in the factory-made serum, and when we mix it with Mr. Smith's A blood, what's going to happen? Nothing. Nothing's going to happen. It's just going to be like swishing around in the bottom of the test tube. We're not going to see any clumping. Okay. Because we don't have the B antigen. We only have the A antigen. Okay. So we said Mr. Smith is A positive, right? Did I say A positive? I mean, I just said A blood type. Let's say he's A positive. All right, Derek, you talked to me about this earlier. If you have the A positive, then that means you are positive for the Rh antigen. If we were to take the anti-Rh serum that's factory made and we mix it with Mr. Smith's blood, it's going to clump, right? Because Mr. Smith has the Rh antigen. All right, so what's his blood type? He's A positive. Now, what antigens are present? He has the A antigen. What antibodies are present? Well, we know naturally occurring, he's going to have the B antibody. All right. So we good? You want to go down to Miss Jones? All right. Let's say Miss Jones is B negative. All right. Miss Jones is B negative. So she has the B antigen, and then she is negative for the Rh. So we use the anti-A serum, mix it with her blood, and we do not see any reaction. It's just swirling around. We take the B antibody and we mix it with Mrs. Jones's blood, and all of a sudden we see a big clump. And that means that, okay, so if we use the B antibody and we mix it with her blood and it clumps, then that means she's got the B antigen. She's got the B antigen who's finding the the B antibody, and they clump together. So we know that we can type Miss Jones as at least B type blood. So if she were to get any kind of transfusion of blood, she would get B blood products. Now, we use this antibody RH, this RH antibody, and we swish it around, and we see that there's no clumping with her blood. Well, that means that she does not have the Rh antigen. Okay, so the interesting thing, when we don't have the Rh antigen, we don't want to give her B positive blood. Why would you give somebody the A antigen, or excuse me, the Rh antigen? So, especially if you're of childbirthing age, if you are at the age where you are able to have a child, or you're still planning on having a child, we don't want to give you an extra Rh antigen, okay? Only in emergency circumstances. And we really try to avoid that. So we will give her the Rh negative blood, B negative blood. So what type is she? She's B negative. All right. Antigen's present. She has the B antigen. And then the antibodies would be anti-B, anti-A, antibody A. Okay. So I guess people are getting that because I'm not getting any response or any questions. All right, Mr. Green. Let's make Mr. Green O negative. Mr. Green is like a very highly coveted individual because Mr. Green is O negative, which means Kentucky Blood Center will be calling Mr. Green every 3 or 4 months to say, Mr. Green, please stop by and donate blood because you are a universal donor. And what that means is that Mr. Green's blood can go into anybody's blood, all right? Because Mr. Green is O negative. Mr. Green does not have the A antigen. Mr. Green does not have the B antigen. All right? Hold on, I got to chat. I'm going to pull this down. For antigens present for Mr. Smith, since he's A positive, does that mean he has the A antigen and the Rh antigen? That's correct, Hunter. Absolutely correct. Spot on. All right. Excuse me. I don't think I've eaten today. Mr. Green, he is O negative, right? We said he's O negative. So he's not going to have the B. We're going to use that factory made serum and we're going to mix it with his blood and we're going to centrifuge it. That's what I mean, like swish it around or spin it. And we're not going to find any clumping at all because he does not have the A antigen and he does not have the B antigen. He also, if we use the Rh factory made serum, he's not going to have the Rh antigen either. Basically, when you get Mr. Green's blood, it's just red blood cells without all the crap that we have to spin out of it or consider for. That's why gunshot wound victims, trauma victims, people who get airlifted to trauma centers, they have O negative blood on deck in the ER ready to go for these people. I mean, this is when it's like life and death, where they won't do any typing at all. They won't do any typing. What these people need, they need a blood and they need it now because they're bleeding out for whatever reason. And they will quickly rush to this little dispenser in the ER. I'm not sure if you guys have seen it, you know, and they may type a code in, all of a sudden the door flies open. It's just like bars of gold in there. And what I mean by bars of gold, it's O negative blood. And they will transfuse, 20 units, 10 units, however many units you want. That's why Mr. Green gets the call all the time, because Mr. Green has to go, because I don't know, they may even pay these people. Sometimes like you can get paid for plasma. I'm not sure. Sometimes they may pay for O negative blood, but you know, he's won the ticket. And the good thing is that O negative blood population, at least around here, I think is still around 30 to 40%. It's pretty high. I know my grandmother's O negative. I'm A positive. My whole dad's side of the family is A positive, so I'm sure that's where that came from. But so to get back to what I was talking about, so if Mr. Green is O negative, then chances are he will have the A antibody and the B antibody because it's naturally occurring. That's just how genetics are. So he's going to be positive for A antibody and B antibody. Okay. And he can still receive O negative blood. Now, he cannot receive B blood and he cannot receive A blood. Because if you think about it, if Mr. Green's got A antibody and B antibody and we try to give him A blood type or B blood type, we got those antigens in there, right? So we cannot, you know, that'll cause clumping in the circulation and we can cause severe damage to Mr. Green. Okay, Miss Brown. Let's talk about Miss Brown. We okay on Mr. Green? It's kind of funny how I named him Mr. Green because it's like Mr. Money Man or something like that because he is the money man. He is O negative, if that helps you remember. Universal donor, Mr. Green. Okay. So we're cool there. Yeah. Yeah, I mean, yeah, that's right, Derek. Means right, he's ready to go. That's right. That's how you remember it. He's ready to go. You can just suck him dry and give him to anybody who needs it. Okay. I think you can deliver. I don't know how many. You can probably donate 2 units of blood, I think, every 3 to 4 weeks. What I mean, you can't just like drain somebody like that. You can probably get, I think it's 3 to 4 weeks, you can come back and that means your blood supply has been raised up enough to where they can take another unit or two from you. Okay. All right. Mrs. Brown. Mrs. Brown. Let's say she's AB. Okay. AB positive. So Miss Brown, if you guys are catching on, this is when I would ask you in person, but it's like I'm staring at a bunch of screens. Mrs. Brown is AB positive. So she's going to have the B antigen and she's going to have the A antigen. And she's going to be the RH antigen. She's going to be like lighting up like a Christmas tree. All right. She's going to have the A antigen, the B antigen, and the RH antigen. Now, she will not have any of the antibodies, right? She will not have A antibody or B antibody. And if you think about it, if you really want to get into it, we can give Miss Brown blood to Mr. Green because Mr. Green does not have any antigens. Okay. And Mrs. Brown has, she has both A antigen, B antigen, and the RH antigen, and she does not have any of the any antibodies. So technically, you can give Mrs. Brown's blood to. No, excuse me. I'm sorry. You cannot. I apologize. You cannot give Mrs. Brown's blood to Mrs. Green to Mr. Green because she. I don't know where I was going with that. Mr. Green's has A antibody and Mr. Green has B antibody, right? You cannot give Miss Brown, who has the A antigen and the B antigen. I'm sorry, that's a mistake. Let's just keep this kind of simple, stupid. We don't need to talk about transfusion anymore. I've already embarrassed myself. So does everybody kind of understand that? That takes up a lot of our time. Please kind of chat me something if you have questions or reach out. Okay. Yeah, sure. Sorry, Hunter. So Mrs. Brown, and we're going to tag Miss Brown as AB positive. Okay. So if she's AB positive, then she's going to have the A antigen and the B antigen. So if we use those serums, those factory made serums that I'm talking about, and we put a drop of that serum into these test tubes with Mrs. Brown's blood, and we spin it, centrifuge it, we're going to see clumping at the bottom. And that tells us that she has both the A and the B, because we use 2 of those different serums, and we see clumping on both anti-A serum and anti-B serum. That means she has AB blood. And then likewise, when we use the RH antibody serum and we see clumping, that means she is positive for the RH antigen. Therefore, she does not have any antibodies, and she will be the opposite of Mr. Green. I think Ryan brought that up earlier. It's just the opposite. Okay. Comprende? All right. Just let me know. So it's going to be in the lab manual, Derek. Can you guys see the lab manual? I thought it was on there. Okay, good. That's a problem. You all should have had that for a while. Okay. Yeah, it's in the lab manual, Derek. We're going to be in lab 1, and you'll kind of see all the labs built in there. It won't be like any individual. The way Bio 139 is that there is no each individual document. It's going to be the whole lab manual, the whole thing. Now, if I post some interesting supplemental stuff on there, then I'll email you out. When we're going to do this, I'm probably going to send it on. I'm going to put it, once I figure it out, I'm going to put it on my YouTube. And then if I can try to upload it to Blackboard, I can. It's all that compatibility stuff with Blackboard. And it kind of gets messy, and people say, "Oh, it didn't look right," and all that. It'll look good on YouTube. I know that because all my spotlights do. So just you'll get an email overnight, and or maybe very soon, and we'll kind of I'll show you. Okay, so I got the chat is blowing up. All right. So O has no antigens and AB has no. Yeah, you got it, Ryan. That's it. Yep. Courtney seconds me, and yes, the lab quizzes will not open until this weekend. If you're a little confused on that, just check your syllabus. You'll probably see that the quizzes open up this Sunday. There's no quizzes because we haven't done anything yet. So I have a Thursday section that I got to talk to these people about, and then I'm going to give you guys a couple days to kind of consume this material. And then on Sunday, I'm going to open up the quiz, and I'm going to have it run through next Thursday. And it'll be a quiz over this material. Okay. Yeah, Raymond, you got it. Thank you for a second, Raymond. What type of what type blood can you give to somebody who is AB? Yeah, you can. You can. Now, I think they can only get their own. They can only get their own blood. Because, and I'm trying to think, actually you can give them. All right, here, we're gonna level up here. All right. When you go to Kentucky Blood Center and you say, I want to donate blood, and you're Mr. Green, and they say, oh God, it's Mr. Green. We got to take you on back. We're going to hook him up. We may put the TV on for Mr. Green and ask him if he's hungry or if he's thirsty, because we want people like Mr. Green because he's O negative. When they pull your blood out, they separate your naturally occurring antibodies and your red blood cells. All right. You're just not getting somebody's like raw blood. They take it back to the people in the back, and they separate your plasma, your platelets, and your red blood cells, and they can create 3 products, just more products, fibrinogen, coagulation factors. They do all kinds of stuff to your blood back there, okay? When somebody receives blood, they receive packed red blood cells, all right? Packed reds, as we call them in the field. And what that is red cells only, all right? So when Mr. Green comes in, he gives red cells only. He's only given his red cells. He's not given any antibodies. He's not given anything else. Only red blood cells. And when that happens, if you remember, since Mr. Green is O negative, he has no antigens. So somebody who's AB positive can receive Mr. Green. Mr. Green's packed reds. All right. Now, if they were to receive Mr. Green's whole blood, which is everything, the whole damn thing, then you're also going to be getting antibodies of Mr. Green, which is antibody A, antibody B. And if Ms. Brown has AB antigens, well, that's a problem, big problem, because now you got 2 of them to deal with. All right, does that make sense? Let me know, Hunter. I got like 12 chats I got to do now. Yep. All types. Yep. Okay. All right, Vanessa, I will check out your syllabus. Okay, it should be a PDF file on there. Try it again. If not, I will email that to you. Just email me later and let me know. Okay, yeah, maybe Courtney can email that to you. Okay. All right, so Clarissa's got some interesting information here. Let me just go review it real quick. Yeah. Blood type AB can receive O. They will not be able to receive A because if you think about it, if you A blood will have B antibody. All right. So that B antibody is going to attack that B antigen on AB people. All right. So I think in this case, they can only have AB their own and then O, not A or B. Okay. All right. So that's a lot of information. I'm probably glad that you guys talked to me in recording this. It'll be a good way to go back and revisit it. If you do not understand this, I don't want you to wig out because this is elevated material, and chances are you may get maybe 2 or 3 questions on this on the test, and you may not ever have to deal with this ever again. Okay, so I don't want to discourage people saying, "Oh my God, this class is going to be horrible." You know, if it's going to be like this the whole semester, I can't do it. Not the case. We started off on some pretty heavy material. Okay, it's not going to be like this the rest of the semester. There's been things in graduate school that I went, I'm like that. I was like, they wanted to go over this stuff. I said, I'm never going to use this. And I did not, I spent an adequate time. If I understood it, I stood it, but I kind of took my chances with it. I don't want to say you want to do that for everything because it does, you know, it'll add up, but I do not want to discourage people who may find this extra complicated and think that this is the whole class because it's not. I wanted to mention that before we move on. Yeah, they probably will. The spotlight will, those guys will probably, they're a lot more seasoned than I am, and it's a different perspective. And I do encourage everybody to listen to the spotlight sessions. Okay. Now, the pig, arteries of the pig. Check that out. That's really not something that I have anything to go over. This is just pure memorization that, you know, that you get a lot of that in your entry level classes. So please review the pig. There will be, when test time comes and I see pictures like this, I will probably refer you back to certain areas of the pig because I don't want you spending too much time on this memorization part, but you will have to know some of this for the quiz. I may even send an email out over the quiz detailing where I want you to focus on. It may be 1 or 2 questions. The others will probably be conceptual. Okay. So when you find your way working through the pig and the labeling exercise, I may be able to get you the answers on page 8 for lab exercise 9. I'll have to see if I can find something on that, see if Mr. Davis has posted it. Now, scrolling through back through 11 and 12, I think 12 gives you the answers to pages 9, 8, and 9. And then 13 is more the same. Okay, and then we have the neck model. This goes on and on. All right, now page 15. 23-year-old BCTC student was in a car accident and was badly cut on the arms and neck. A passing state trooper stopped to investigate the incident and pulled the victim over on the overturned car. He noted a pulsatile bleeding from a wound. So if you have pulsatile bleeding, that means it's pulsing. That's your heart contracting, and that causes a pulse-like bleed, which would mean that you have struck an artery. And the victim's left arm and a steady flow of blood from the wound of the neck. Okay, so I want you guys to work on these yourselves after you have worked through the memorization part of the pig. This is basically focusing on arteries and veins. okay, and try to answer these questions. I don't think they're going to be too hard. The pulsile part is going to be the arterial blood, which is oxygenated blood, which will be probably like a bright red. And then if you've ever had your blood drawn in a tube, you might recognize that it's, ooh, this is dark blood. It looks really dark and maroon-like. Well, that's because it came from a vein and it's deoxygenated blood. So that's just something kind of interesting. But work through these questions. I think you will, if you have any questions, please reach out to me. But I kind of want you guys to work on that on your own when you move through the vessels, memorizing them. Okay, so let's see. Yeah, quizzes are timed. Everybody answered that. I will probably be a little bit lenient on that time. It's supposed to be 10 minutes. I may give you 12. We'll just see. All right. So that is all we're going to cover today. It's a lot of material. Please check out. I do probably encourage the spotlight sessions. Please do that. They may go a little bit more in depth or explain it completely different than me. And that may click with people. And as long as you get the material and you understand it, then that's what we're here for. So if you do feel confused or if you are confused, please reach out and we'll be able to provide you some clarity. But okay, hold on. Yeah, Liz, let me, yeah, that's 24th. Yes, ma'am. It's going to open up sometime on Sunday, probably around 5 or probably sometime in the evening when I get my act together. Okay. Any other questions from the chat, or does anybody you can talk over your microphone? I'll probably try to post this on YouTube and I'll let you know the details on that and we can kind of go from there. You may be required to attend X amount of them. I know in Bio 137, that's what they're doing, but double check. It's going to be found in the syllabus. If any of you all have problems with the syllabus, please email me and I will figure out what's going on there, but it should be on Blackboard. All right. Okay. So we're good, right? Chat me something. We okay? Yeah, 6 is mandatory. Okay, there you go. All right, so you guys are good on, okay, all right. Siobhan, I think it's Siobhan, perhaps. I'm sure you'll let me know. Okay, Edward. All right, cool. All right, so I'm gonna turn you guys loose. All right. Yeah, follow your if you get it. Yeah, if you guys talk amongst yourselves, I'm pretty sure that's what happens. All righty, I will send you out an email and we'll talk to you guys later in the week. Have a good one.